Okay. Okay, so I think we can start now. There are a few people on YouTube and also here, and I hope that after in a few minutes more people will join. So welcome everyone to this session of Pratidhwani. So it's a lecture series organized by the Department of Chemistry, IIT Delhi, and welcome you all and a very good afternoon. And today we are lucky to have with us Professor Sourav Bhattacharjee from University College, UCT Dublin, University College Dublin. So before the start of the lecture, let me give a brief introduction of Dr. Bhattacharjee. So Dr. Bhattacharjee was born in Kolkata, where he also received his schooling. He joined the Medical College and Hospital, Kolkata in 1997 and graduated with an MBBS in 2004. After a brief period of resident training in orthopedic surgery, he joined MSc in Biomolecular Sciences, Cell Biology at the Vrije University of Amsterdam. In meanwhile, he moved to Edinburgh, UK, where he conducted research on the toxicity of nanoparticles under the supervision of Professor Vicky Stone, who is from Napier University, Edinburgh. And then he began his PhD at Wageningen University in Netherlands in 2008, uh, where he successfully defended his thesis in 2012. Following that, he worked for a year as a postdoc at Mesa Institute for Nanotechnology, University of Twente, Netherlands. In March 2014, he joined Professor David Braden's group at University College Dublin in Ireland as a postdoc working on the EU FP7 funded Trans INT consortium, trying to develop nanoparticulate drug delivery platforms for oral insulin delivery. From February 2016, Dr. Bhattacharjee joined the UCD School of Veterinary Medicine as an assistant professor of anatomy. His current research interests include developing advanced theranostic platforms uh, using biophotonic and in silico tools. Professor Bhattacharjee continues to be a registered physician with the Medical Council of India. So a very warm welcome, Professor Bhattacharjee, and over to you.
Thanks, Shomik, and welcome everyone. I hope you can all hear me well, and if not, just flag it in. First of all, thanks a lot for this incredible opportunity. I mean, I have never met you guys, but I've always felt that I need to meet you, and I think this digital world gives me this opportunity to actually catch up while sitting down in different continents and different time zones. So I think it's an incredible opportunity again for me. Thanks a lot. And before I even begin and share the screen with my slides, I thought when Shomik came to me and said, okay, I have to give a talk, I was rather like, what shall I say after 5 o'clock or 5 p.m. in India, because it's typical after work time. So rather than trying to giving you a deluge of data, I thought it will be rather pragmatic to give you some of the stories of my life and what I have learned so far working with nanomedicine, and trying to give you a kind of a treat and homage, if I may say, regarding the different chemical or the chemistry stuff and perspectives that I have received over this course of my life and career. With that, let me share the screen first. Okay, so I share the screen. That's it. So just a moment, it's sharing. Yep. So you should be able to see this. Just a moment. Yep. I hope you can see the screen. Do you, Shomik? Is it all right?
Yeah, it's perfect.
Fantastic. Let me start the laser pointer. You should be able to see the laser pointer moving over now. So I would like to welcome you once again to my story today, that is negotiating barriers in human body with nanoparticles and role of surface chemistry in drug delivery. And before I even begin, I will warn you in advance that I'm going to pester you throughout this presentation with this word surface chemistry over and over again, because I think the importance of surface chemistry in developing nano formulations, particularly say from the perspective of oral drug delivery and cancer nanomedicine, is often underrated, and it needs to be emphasized more and more. So today I just assembled a kind of a story for you, talking about the different surface chemistry and how it actually influences the drug designing which we chemists should actually encompass within our research modules. But before I start with this story with surface chemistry, maybe it's time to just give you a quick career trajectory of mine so that you know, before surface story, what is my story actually. So as Shomik has quickly summarized, but I would like to go through once more. In 1997, I entered the Medical College, Calcutta. I finished my MBBS in 2004, so somewhere around here. I did a quick residency in orthopedic surgery. And then from 2006 to 2008, I got admitted into MSc in Biomolecular Sciences with majors in cell biology in the Vrije Universiteit Amsterdam in Netherlands. While my thesis work was mostly done in the Napier University, Edinburgh, Scotland, which I finished my MSc in 2008, but got quickly imbibed into a PhD program in the Wageningen University, and I finished it from 2008 to 2012. After that, I did 1 year of postdoc in the Mesoporous Institute of Nanotechnology, part of University of Twente, Netherlands, and I worked on a microfluidics project trying to develop organ on a chip. And then from 2014 to 2016, I worked as a postdoc in University College Dublin under Professor David Braden. And we were part of a large EU Framework Program 7 consortium, which had partners from all over the European continent. And we were working in a program called Transient, where we were trying to develop oral nanomedicines for insulin delivery in diabetes patients. From February 2016, I'm working as an assistant professor in University College Dublin. And you will be surprised that I now even teach anatomy and also do research which encompasses anatomy, evolutionary biology, and nonetheless, I keep on researching on nanomedicine. So you can just say that I'm pretty much of an academic nomad, and I'm rather driven by inquisitiveness rather than going into department to departments. Now, having said that, first introduction to the field of nanomedicine, and this is some of the beautiful frescoes from my favorite city of Edinburgh. This is where I actually was kind of baptized to the field of nanotoxicology, and then which ultimately evolved into nanomedicine. And Professor Vicky Stone was kind of my mentor. She brought me into this field of nanotoxicology. And that is where I actually began working with nanomedicinal stuff as well. And regarding during my PhD tenure as well, I quickly got involved into also the chemistry part, and I tried to synthesize my own nanoparticles. I characterized them both photochemically and physicochemically. And from then on, I have continuously stuck around with this nanomedicinal, particularly from drug delivery perspective. So what are the promise of nanomedicines? I mean, nanoparticles are unique. They can deliver different things. They have unprecedented properties, which bulk materials are not able to deliver. And over the last couple of decades, if you look around, there has been a complete magnitude and a wide plethora of different nanoparticles that have emerged with drug delivery and different other medical management, including theranostics being involved. Just to give you a quick glance of these different prototypes of nanoparticles that are being currently used, we have the liposomes, micelles, solid lipid nanoparticles, polymersomes. So there is a huge range of lipoidal nanoparticles that are being used. We also have different polymeric nanoparticles, dendrimers. Now, dendrimers are my personal favorites. There are different reasons we can discuss during the Q&A session. We have the hydrogel materials. Now, hydrogel is typically developing as a biomedical scaffold type of proteins, but still some nanoparticulate formulations are possible. We have mesoporous nanoparticles. I have personally worked with some mesoporous nanoparticles where these particles are well defined, but at the same time, they carry different pores, and these pores then can be loaded with different drug materials, and then the drug materials leach out of these pores and giving you a targeted and sustained delivery. I have worked with porous silicon nanoparticles as well. These are, again, beautiful scaffolds and the very porous, of course. And these pores can be, again, loaded with different biomacromolecules. We have different gold nanoparticles. These are, again, my personal favorite because you can get them in different sizes. In fact, nowadays, they are commercially very much available. They are very affordable. The surfaces can be conjugated with multiple different molecules, making them very target-oriented, which is, of course, a dream game for we formulation specialists. We have worked with the different Buckminster fullerenes, for example, the C60 buckyballs. We have the carbon nanotubes. These are the typical 2D materials in form of the graphene sheets. And this is the new generation of nanoparticles coming up and evolving. And I'm very hopeful, and I have a lot of expectations from this particular prototype, which are the nanocrystals of the drugs. I mean, in the Q&A session after the talk is over, I will be very happy and delighted to engage in a discussion with you regarding the nanocrystals, given that I have a lot of hope for this particular type of emerging prototypes. But today, our talk is mostly about the physiological barriers. And when I say physiological barriers, I will take a kind of a more liberty, and I will also give you an example of a pathological barrier. Now, what are physiological barriers? Now, these are barriers which has inherently developed, or rather I should say evolved, in human body over millions of years to safeguard us from the different external pathogens, exogenous particles, including nanoparticles per se, different allergens, for example. And these physiological barriers are extremely important because somewhere down the line, they kind of provide with a threshold for which external pathogens or harmful substances to our body cannot or should not cross. Now, there are different physiological barriers in our body. We cannot go through all of them. But during the process of nanomedicine development or the evolution of this field, two physiological barriers have become very important because they are kind of a mammoth challenge for the nanoformulation specialists to overcome. In this talk, I'll give you an example of two physiological barriers and one pathological barrier, just to give you an example, the kind of challenge these barriers actually throw to us when we work in the laboratory. But even before we start discussing the barriers, it is very important that as a chemist, you already kind of train or spearhead your ideas or your thoughts towards the surfaces of these barriers and what are actually we trying to negotiate with our nanoparticles. Because the nanoparticles don't see barriers, nanoparticles see surfaces. And that is where I would like to kind of promote this idea that you don't see barriers per se, but what you actually see are the different surfaces and the complexities, I would say rather inherent complexities, they present for a formulation specialist while trying to develop nanoparticles that would be able to engage with these barriers, cross these barriers, because these barriers are not very friendly or permissive towards the nanoparticles. And they are like that because evolution has made them developed or has developed them like that for over millions of years. So they are incredibly formidable. To cross these barriers is very difficult with particles. Let us start with the gut mucus layer. The mucus is a physiologically relevant hydrogel, which is heterogeneous, and it covers through the gut, intestine particularly, in humans. Now, we have mucus in different parts of our body. For example, respiratory tract, we have a mucus coating. Female reproductive tract, there is mucus. Even in our eyes, we have mucus. So mucus is a very formidable and extremely efficacious hydrogel layer covering or bathing the epithelial layers of our body. And especially the gut mucus layer is tremendously important from the perspective of developing oral nanomedicines. This mucus hydrogel, it has a lot of water. In fact, by weight, more than 90% of it is water. And the major solid component comes from the biopolymer called mucin. This mucin is like a fibrillar network. If I may compare it, it is like, for example, the agarose gel, what you prepare typically in your labs while doing electrophoresis experiments. And they provide a kind of a sieve to trap the nanoparticles. And this mucus is inherently kind of recycled within our human body every 2 to 4 hours, for example. And these trapped nanoparticles are then also gone for the good. It is recycled within the body with that mucus. Now, it brings a huge challenge, because that means if nanoparticles are adhering to this mucus and then getting kind of recycled or even metabolized, that means we are losing a kind of a big fraction of the dose that we have already administered. That's a huge loss. Secondly, from a pharmaceutical point of view, it brings a tremendous challenge, because, for example, in case of diabetes patients, we need to have a full control of the dose that we are administering to the patient. And in case we are losing certain parts of our nanoparticulate dose or nanoparticulate load, that means we are also losing insulin. Now, that has also other implications. Number one, that means the diabetes patient will not have adequate insulin, so the therapeutic relevance of the dose that we are providing will be curtailed. And B, the production costs will actually grow. Now, let us look into the mucus layer into more detail. The mucus layer, it is a heterogeneous layer. So this is the epithelial cells. You can see it clearly. Now, this is the intestinal epithelial cell layer. This is the luminal part. So here starts the lumen. And just on the surface of these epithelial cells, we have this mucus layer. Interestingly, mucus is not a homogeneous layer. It is a heterogeneous hydrogel. And on certain parts of intestine, they are bilayered. In fact, for simplicity, in this cartoon, we have depicted it as a kind of a bilayer. You can clearly see there are two layers. One layer is here, which is kind of more grayish, that is adhering to the epithelial cells. In fact, the epithelial cells, they secrete this mucus. This is kind of more thick, and it is closely adhering or attached and more organized because it is attached to these epithelial cells. This layer, which is adherent towards the epithelial cell, they have a lot of immunoglobulin A, and that is why they are very important for our body's immune system. And we have a lot of antimicrobial proteins. Towards the luminal side, the mucus layer slowly gets more disorganized, loose, and fluffy. So this is the outer mucus layer here, and there is a humongous range of microbiota and population over there. Now, these are very important commensals for our well-being. So mucus, overall, if you look into that, it takes part into our body's immune system. It contains some very important antimicrobial proteins, which we need to safeguard our body from different pathogens, and it has very important population of commensal microorganisms, which are very important for homeostasis and the well-being of us. But the problem is, mucus also creates a barrier for our nanoparticles, because we want our nanoparticles to actually cross the mucus barrier. If it doesn't cross, it will not actually reach the epithelial cell layer, and then we have a problem. Because our aim is to actually cross the mucus layer, reach here, and hoping, or we can speculate, that this epithelial cells will actually uptake these or take over these our nanoparticles, and hopefully then the drug will be released into the bloodstream. But that is not what actually happens mostly. Now, there are some particles, and you will be surprised, and due to the crunch of time, I will not be able to go into very detailed discussion about this type of nanoparticles. And these type of nanoparticles, you will be surprised, are mostly viruses. Now, viruses, with their typical diameter less than 100 or equal to 100 nanometer, can be often considered to be nanoparticles, and we call them viral nanoparticles or viral particles for simplicity. Now, it is extremely important and rather staggering, I would say, that viral particles, they're often able to cross this mucus layer as if almost they are freely diffusing in water. And we still don't understand why. The problem with nanoparticle is you can have basically two types of interactions. One, there is a steric barrier. So some particles feel the steric hindrance and they are basically repulsed back. And also there are some interactive barriers. So some of the particles actually permeate through the mucus layer. But inside mucus, we have a kind of a network of these mucin fibers. I am showing it here. And these nanoparticles, for example, here, they are trapped into it. So this mucin network rather acts as a sieve or filtration mechanism where these nanoparticles are engaged and trapped and then again recycled. So this mucus layer is actually recycling, and the adhered nanoparticles are then just simply wasted. Now, you might ask how these nanoparticles actually adhere. Well, first of all, this is a network, so sometimes the pores are not simply big enough for making these nanomaterials or nanoformulations to permeate. Other than that, these mucin fibers have different cationic and anionic sites. Now, under physiological pH, the number of anionic sites is more than the cationic sites. So, in under physiological pH, again, overall mucus is anionic, and the mucin network is anionic, to be more precise. So, the nanoparticles bind to the cationic and anionic sites depending on its surface charge, and that acts as a catalyst towards binding of nanoparticles and trapping it. So again, I think I am very clear here. This surface chemistry, especially the surface charge, and how these particles negotiate with the mucus or the mucin network becomes extremely important. And I would like to engage with you further during the discussion sessions, what are the new paradigms that are emerging and the new kind of strategies we are devising right now to produce nanoparticles or synthesize nanoparticles that are able to not only negotiate or engage with this mucin network, but rather also able to permeate it. Because if we are not successful here, then the future or translational hope for oral nanomedicines look rather green. So we have to cross this mucus layer here, but the key lies in surface chemistry, and we are working on that. And that once again shows the importance of surface chemistry while designing nanomedicines. Now, you might be thinking, oh, it doesn't do, what about this epithelial villi? In fact, this looks pretty simple in these cartoons, and this is kind of my own perspective that the simple cartoons in the textbooks are not at all close to the reality. The reality of intestinal villi is somewhere like this. So this is in small intestine, and the mucus layer is just stripped off. And you can see this kind of this intestinal villi, and I hope you already realize this humongous amount of surface area. This intestinal villi actually forwards to the nanoparticles. And once you are putting the nanoparticles, even if it is crossing the mucus, it is now coming in contact with this very high surface area where it can stick. Now, it is very difficult to predict how the nanoparticles will actually behave once it comes or exposed to this very high surface area in the intestine. And I hope as chemists you are already on my side to agree that this is a kind of a big challenge while developing nanoparticles, because first we have to negotiate the mucus layer, and now we have to go through this entire surface area of contact through which the nanoparticles has to pass if it has to exert any therapeutic effect. Now let's talk about another physiological barrier. This is a very important barrier. We call it blood-brain barrier. And this blood-brain barrier actually safeguards our central nervous system, most importantly the brain, from different external pathogens and microorganisms. The issue is blood-brain barrier also stops nanoparticles to actually reach the brain, which is kind of good because brain is of course very important organ, and we don't want to get it exposed to different external or exogenous agents. But at the same time, it creates a kind of a dilemma, because the nanoparticles or the nanomedicines that we produce, we want them actually to reach the brain, because otherwise how can we get the therapeutic effect? Now there also surface becomes very, very important. Before I go there, let us quickly go through and try to understand what is the blood-brain barrier, aka BBB. So this is kind of the brain. You can see it has all the meningeal arteries. And then we go with this. This is typically an artery or a small branch of an artery. And we will be able to see the astrocytes, which has this big pores. You can see these astrocytes over here. Now, these are typical kind of a star-shaped specialized cells. And these astrocytes, these podocytes, actually, they go over here. And you can see they're merging with the blood vessels with the epithelial layers. Now, if I have a cross section here, so this is the blood vessel, and you will see the end feet of the astrocytes are kind of encircling it or wrapping it, acting almost like a cloak to cover it from all sides. These junctions are very tight. So what actually means is, even if you are able to reach through the bloodstream to the brain with your nanoparticles will be pretty much encompassed or surrounded here, and there will be very little scope for the nanoparticles to go through or leach out or extravasate through these blood vessels. Because first of all, if the nanoparticles have to extravasate out, which is essential for it to exert any therapeutic effect, first it has go through this epithelial layer, but then it faces this extremely strong barrier due to the end feet of the astrocytes. So it becomes very difficult for the injected dose, because I'm using the word injected because these nanoparticles, unless otherwise mentioned, they are always given or administered intravenously. So the injected nanoparticles find it extremely difficult to permeate through these both layers and then come out. So for example, there is a brain tumor developing in this F cartoon. So this is, for example, there is a tumor and the tumor cells are developing over here, but your nanoparticles are unfortunately kind of crammed up in these blood vessels. And to actually go through and actually reach the tumor to exert any therapeutic effect and hopefully cure the tumor cells, they need to cross this blood-brain barrier, which these podocytes and the end feet of the astrocytes are creating a lot of issues, and they are actually barring it from accessing the nanoparticles from accessing the tumor tissue. Nowadays, we are working with different liposomal formulations, for example. Typical lipoidal other formulations are also available. You can see there is an encapsulated drug payload, and by different surface functionalization, people are trying to target different surface receptors and hoping that the cells will be taking up all these liposomes, and that will enable these nanomaterials to cross the blood-brain barrier and finally release the drug. But once again, this blood-brain barrier, the way we are depicting in the cartoons, they are pretty simplified versions. The reality, unfortunately, is this. You can see this is the kind of a realistic rendition of blood-brain barrier. I think it is very clear that humongous amount of surface area. Once again, this red part is the arteriole, so the nanoparticles will be inside it after you administer it intravenously. And once they leach out, if they leach out, you will be seeing all these astrocytes here, and they will be having all these end feet circling it. So just try to imagine this incredible surface area. Even if these nanoparticles just leach out or extravasate out of this vessel, they have to now negotiate with this humongous area, surface area. And there is every possibility that the nanoparticles that you have prepared after working very hard in your lab for weeks or months or years, they will be lost somewhere. And these are the kind of the challenges. The surface story continues. Now I will now present with you an example of a pathological barrier. And when I say pathological barrier, it's kind of different, in my opinion, from typical physiological barriers. I'll not go into detail too much, but I am sure you have heard about the tumor microenvironment. In a developing tumor, the internal matrix or the parenchyma of a tumor is hypercomplex. There are different types of cells. Let's not get into there because that's not the aim of this talk. Rather, what we are trying to understand as bunch of chemists is the complexity of the surfaces. So this is kind of a tumor developing. Tumor matrix is visible. It's kind of spherical, duffy in consistency, kind of tough. But let's say this tumor is pretty much on the earlier stage of its development. These are the blood vessels. You can see the RBC is circulating over here. The blood vessels get in. So if you're injecting intravenously your nanoformulations, let's speculate that these RBCs are not RBCs. In our case, these are nanoparticles. Now, the nanoparticles will actually get through the tumor masses, but then it needs to come out. Because if it does not come out, it will not be able to negotiate with all these different cells that are present in the tumor microenvironment. And unless it can do that, the nanoformulations are rather ineffective with very little or no value. Now, the problem is, if you look into this tumor microenvironment, there is a humongous amount of collagen material, extracellular matrix, different types of cells, even different types of biomacromolecules, and you want to precisely target certain areas with your nanoparticles. So somewhere down the line, it becomes almost trying to target finding, or it's as tedious as trying to find a needle in a haystack. And that is where the major challenges are. Now, how complex is this network or this tumor microenvironment? Once again, this is a simplified view. The reality is something like this. And these are the typical intratumoral arteries, and other venules are not shown over here. You are putting your nanoparticles. These are the different cells. There is an incredible amount of fibrinous materials. You can see these fibrinous materials over here. There is tremendous amount of basement material, different cells, which are shown over here. And tumor is extremely heterogeneous. So you're dealing with a very complex matrix, which is absolutely heterogeneous, absolutely unpredictable. You cannot model or mimic it. And that's where, with this incredible surface area also available to actually attach to your nanoparticle, and most of the times to kind of make it invalid. In fact, surface chemistry is also it also puts a challenge from especially intravenously administered nanoparticles. So for example, this is a human body. You have put some nanomaterial in intravenous administration. Now, these are your nanomaterials, for example. You have functionalized it with different ligands. Just as for an example, I have shown some antibodies. These antibodies will be circulating in the blood vessel. The issue is these blood vessels, once they enter the bloodstream, these nanoparticles are notorious for engulfing or getting surface adsorbed with different proteins, which has a kind of a connotation called opsonins. These opsonin proteins, they get surface adsorbed on these nanoparticles and make them kind of very tasty or palatable product for the phagocytes. These phagocytes quickly engulf them, and then these phagocytes go into the reticuloendothelial system and the different organs, for example, lungs, liver, spleen. So what happens is, and I have seen it actually, this accelerated blood clearance, or what in short ABC of nanoparticles, this phenomenon is like super fast. It's brisk, it's rapid. And even before you can measure anything, in most of the cases, 90 to 95% of the injected nanoparticles are already sequestered or already being phagocytosed by the different macrophage cells, and they immediately get kind of refuge in lungs, liver, and spleen. So imagine you have produced nanoparticles, you have worked very hard, your nanoparticles are great, you're very happy as a PhD or a postdoc or any researcher of any field, and then you inject it, and within seconds, 90% of the nanoparticles are just gone for good. I think that's very frustrating. But what actually is happening is the kind of the major antagonist is happening over the surface. The surface attracts those opsonin proteins, and that creates this mess where most of the, in fact, 90% or more, sometimes even up to 99% of your dose is actually gone. And that makes an incredible challenge for a formulation specialist, because if you are losing 90% of your dose, then it's difficult. So nowadays, we are trying to kind of work around with the surface chemistry, because the hypothesis is we can stop this type of phenomenon like ABC of nanoparticles by carefully engineering the surfaces of it. So this is just a cartoon showing you a liposome. And liposomes, I have given the example of liposome because liposomes are the most successful translated nanomedicine so far. So in liposomes, we can kind of decorate the surface with different things. For example, we can put some antibody, we can attach some imaging agents, some targeting ligands, we can put some spacer, add again more antibodies, molecules. We can even give some hydrophilic chains, for example, polyethylene glycol, and we call them stealth liposome. We call them stealth because the idea is attaching these hydrophilic chains on the nanoparticulate surfaces enable them to evade this reticuloendothelial system, and it really ameliorates the accelerated blood clearance, which I shown in the previous slide. So surface functionalization of nanoparticles is the key. At least that's what we have understood so far. Have a nanoparticle, start grafting different things on the surface, because if we can control the surface, we can control its behavior at the nanobio interface. And nanobio interface can be extremely complex. Continued interest in physiological barriers and pathological barriers like tumor microenvironment has thus continued. Have we succeeded a lot? I don't think so, but I will show that in the next few slides. But what is extremely important that we kind of comprehend the importance or the critique of these physiological barriers. These barriers are there to safeguard us, so we need them. But as a formulation specialist, when we are working with nanomedicine research, we want to negotiate them, we want to permeate them, and we want to go across them, because that's where the crux of the success of nanomedicine research lies. We fail here, we fail everywhere, because that is why a lot of interest has been focused now in crossing the physiological barriers. But it's a careful balance. If we compromise the physiological barrier with our nanoparticles, we have a different problem, because these barriers are there. Nature has created these barriers because of reasons. We need them. Once compromised, if compromised, we will not be able to maintain our homeostasis, and we will be sick immediately. So it's kind of a balance which we still don't know how to actually strike. We are trying, and I think in the next couple of decades, that will define how nanomedicine will succeed, or if not succeed at all, or partially succeed as a discipline. Surface chemistry is very important, not only for oral delivery. I've given you example of mucus, but there are other issues as well. For example, delivering genetic materials. I mean, DNA, RNA, these are extremely charged molecules. It can bind to different surfaces, especially nanomaterials are extremely reactive, surface reactive, I would rather say. So these are incredible and notorious materials in fact, interact with different nanoparticles. So it's not easy unless you can actually exert a control over the surfaces. Same for in vitro work, and same for in vivo paradigms. What is important to realize here, unless we can control the surfaces, we will never be able to control all these different experimental platforms. And unless we can conclude or we can control these experimental platforms, we will never have enough confidence on the different data that we have before putting a nanoparticle into clinical trials. So that at the last key message, before I move on with certain conclusions, it is very important that we need to understand the surfaces and interfaces. We need to understand its intricacies. Probably should also rely a bit more on in silico tools. We need to understand the mechanisms that drive it. We need to focus what happens when two ligands actually come in vicinity to each other, and once they juxtapose, what kind of actually engagement happens. Do they bind? Do they don't bind? If they are binding, what are the chemical species that is actually driving this binding or not binding? What is happening? And I think unless we understand this transition, it will be very, very difficult to produce nanomaterials or nanoformulations that are successful, efficacious, and translatable. So surface chemistry is very crucial. Whatever we prepare in the lab, whenever we put inside human body, all interactions are happening or beginning at the surface. Unless we control that, it will be very difficult, if not impossible, to actually control the entire fiasco or the series or battery of events that will be following it. We need to understand that nanobio interfaces are hyper complex. The simple cartoons that we find in textbooks are just overtly simplified. They nowhere actually represent the complexity of nanobio surfaces. For example, I have given you that this is the kind of a real-life rendition of a dividing cancer cells. Now just look into the surfaces, just look into its rugosity, look into its roughness. When you are putting your nanoparticles, in touch with this. This is completely heterogeneous, hyper complex. You have to negotiate with these surfaces. In fact, in cancerous cells, you have particularly no idea about the mapping of the different receptors. But the problem is, you are dealing with a nanobio interface which is hyper complex. But sometimes your particles also have a very complex surface. So if you typically look into the different nanoparticles, you will find the surfaces are not actually very smooth type that you typically draw in exam or find in different textbooks. Rather, the surfaces are rough, and if there are conjugated ligands on this, or it is a functionalized nanoparticle, the surfaces become even more complex, sometimes unpredictable. So you have already a complex surface material, and you are dealing with even a hyper complex surface. That is where the surface chemistry is the key. I know it is difficult as a chemist and having more than 1 decade of experience in preparing and synthesizing nanoparticles, how difficult it is to exert control at a nanoscale over the surfaces. But that is where the crux lies, in my opinion. Unless we can get hold of these surfaces, we'll never be able to get into a good translation. And that is why, so far, our success has not been great. And that is why, if you look into nanomedicine research, there are plenty of publications, a lot of research being published. Everywhere I hear a hype and hope about nanomedicine. But if you look into actually how many products have been approved by US FDA, the number is somewhere still hovering around 40. And we can discuss about this. There is a high attrition rate of nano formulations. In fact, most of them are failing in phase 3 of clinical trials. In fact, most of them are failing in phase 3A stage of clinical trials. Maybe during the discussion, I would like to engage with you and ask your opinion. Why do you think that is the case? But the truth is, the nanomedicine research, as far as proper products, pharmaceutical products, have not given us the success or hope that we actually looked for. And if you look carefully into the market right now, except Doxil and Abraxane, you don't actually have products which are commercially successful or dispensed regularly in a hospital setup. That's only 2 actual formulations. We have 40 approved formulations, but not much more is coming. And even if they're coming, they're commercially not viable. Now, I have talked about the problems, so I would like to finish up with what are the solutions. Now, I don't know what are the exact solutions. If I would have known, I would have done it myself. But I just can give you my perspective. I think collaboration is the key word. We have zoomed into our disciplines too much. I think it's time to zoom out a bit. We need to collaborate with different people. I mean, the chemists need to collaborate with the biologists, or probably physiologist is a better term. And physiologists should turn or work with the pharmaceutical specialists or formulation experts, because unless we collaborate, we will not be able to understand the depth and the intricateness of this complex surface chemistry driven problem that is at our hand. So collaboration is our key, and I think we can make really a lot of improvement in this field, and we should do that. I think it is extremely important that we define our surfaces. I think what I have tried over the last half an hour or so, I have tried to convince you the importance of surface and the surface chemistry. So before you go to your benchtop with your experiments, define the surfaces that you are working with. Because what you see is these ripples of the surface as a cartoon in your screen that will actually define how the particles will behave inside a human body or any nanobiocomplex interface per se. Get back to basics. I think we are often trying to go ahead of the carriage. Let's not do that. Get back to basics. Let's go back to the fundamental pillars of science where we have learned how to do research. It is very important to get inspired by nature, because what I have seen personally, that nature has solved many of the problems that we are still fighting in our labs. There is no shame in learning from nature. Nature is full of patterns. Nature has solved many of the incredible problems that we cannot even think or dream of solving in our benchtops. So I think that is very important, that we have to be humble student of nature, go back, find patterns. For example, there are patterns everywhere. Now, this is simply a cartoon of nanoparticles. You can see the nanoparticles, they're binding with the different receptors. It looks a nice pattern. But when I'm kind of a more physical and spiritual or even philosophical, I sometimes look for these patterns, and I see these type of patterns are everywhere. In fact, nature has developed all these patterns throughout us. It's just on us. The onus is on us to actually look for them and actually then involve it or apply it in our research. For example, this pattern, what about this? I see incredible similar patterns. And these biosurface patterns are spread across our universe. My own feeling or opinion is we don't look for them sometimes. We don't spend enough time with nature to look for the solutions or the designs that it has already produced us. And I'm sometimes scared to see or apprehensive to see that we have confined ourselves too much into this lab work. We need to go to the nature. Finally, with this amazing surface story, again, that wrote a different chapter in the history of mankind, I would like to thank you all for listening to this simple parable, which I call a surface story. Thank you, and any questions?
So thank you, Professor Bhattacharjee, for the fascinating talk, very colorful and very insightful. And I think people here, all the participants here will appreciate like why there is a huge gap between, the number of papers published basically in this nanoparticle drug delivery area and the actual number of therapeutic agents, nanoparticle therapeutic agents, which are out there in the market. So there is a huge disparity, and I'm sure that we are now able to appreciate like what's the exact difference, like what's the exact reason for this disparity. And we also learned about the importance of surface, and it's a very, I think, uh, it's a kind of viewpoint coming from a doctor like Dr. Bhattacharjee himself. Like, so you have to have things working in the clinic, right? Otherwise, there is no need of
Otherwise,
doing
it's
any kind of
nice,
work.
but
Yes.
unless it actually gets dispensed
Yes.
through the pharmacy,
Yes.
and I know I mean, it's only to me that I'm convinced that most of the nanomedicinal formulations, once translated, it will be translated for parenteral administration, or most of them will be for parenteral, intravenous administration per se. So, for example, Doxil, Abraxane. Now, these are the two kind of prototypes that are given as examples for the success story of nanomedicine. Well, it can be debated. Let's not get there. But if we take for a moment, for argument's sake, that these two nanomedicine or nanoformulations are successful, there is no doubt they are successful. They are both parenteral formulations, by the way. You have to inject them. So oral nanoformulations, I think still we are in a pretty much in a nascent state. We are still learning a lot. And unless we can actually compromise is probably a bit invasive word, but unless we can develop paradigms where we can cross the gut mucus or intestinal mucus in a non-invasive way, I think the chances of, kind of propelling towards more brighter horizons will be difficult. I'm not saying everything is possible. Nothing is impossible in science, but it will take time. I think that's the reality. But at the same time, we have learned new things. And I give the typically example of laser. Laser, when it was discovered, not many people thought it would have any applications. But after a gap of few decades, today laser is one of the most successful discoveries in the field of physics with a lot of medical applications. So far what we have learned, we need to cross these physiological barriers, and that negotiation begins at the surface. Now, you have told me about, of course, there are a lot of papers and publications and reports and MSc theses, PhD theses, postdocs, but how many of them actually you see that focused on the surface? I mean, this is not a criticism. I'm not accusing anyone. This is a common observation and my analysis that most of these articles or these scientific reports which are in public domain, I find them dealing with fancy designs, encapsulation of different materials, and they work in lab. But something working on benchtop doesn't make it translatable to bedside. So we have tons, I don't know how many thousands of nanomedicinal prototypes in articles, but so far, at least till last year, the total amount of drugs that has been approved by US FDA is somewhere around 40, plus minus 3-4. And something is approved doesn't mean it's successful. Approval only means it has to be marketized. Many things are approved, but then they are not commercially successful. For example, inhalational insulin. Insulin, people thought we will have injectable insulin is not easy. Patient compliance is poor. So many people thought that we will have inhalational insulin, which will be just taking a dose through a nasal route. And they were marketed products, but they were not commercially successful. The number of prescriptions were very low, and unfortunately, Pfizer and Sanofi had to discontinue them. But they are great products, by the way. But sometimes it is difficult, even if things are great. People have to buy it.
Yeah, absolutely.
Any other
Well,
questions?
yeah, so I think if you raise your hand, I can unmute you and then you can ask your questions. So meanwhile, we have a question from YouTube. The question comes from Dilip Kumar Mishra. So he's asking that when nanomedicines are coming to the market, like what's your opinion on that? Like
When?
yeah, like
Well,
when?
they're already in the market.
Okay.
I mean, Doxil, Abraxane, I don't know about in Indian perspective, but in Western perspective, Doxil is regularly prescribed. It's a liposomal formulation of doxorubicin. And doxorubicin is a very popular anthracycline derivative anti-cancer drug. It's an encapsulated product with a liposomal membrane, and it works well. But here also, it's not a criticism; it's just an observation. I mean, if you look into now, if you ask me a simple question, and maybe Dilip Kumar Mishra ji, he will also agree with me. I mean, we have all the right to ask that we were giving doxorubicin intravenously. Why shall we now give a particulate formulation of doxorubicin? What is the advantage? Now, if you look into doxorubicin, doxorubicin is very effective drug. That is why it is still in circulation. People use it for different, especially for solid tumors. But the issue is, doxorubicin is a toxic drug as well, very toxic. And often you create more issues for the patient by giving doxorubicin rather than the actual issue or the tumor that you are trying to cure. Now, one of the major side effects or adverse drug reaction of doxorubicin is its cardiotoxicity. It's toxic to the heart. So typically patients have cardiac problems. The interesting feature is for Doxil, which is a liposomal formulation of doxorubicin, it maintains the efficacy of doxorubicin, but it actually curtails the side effect of doxorubicin, especially cardiotoxicity. So Doxil is successful not because it is not more efficacious than native doxorubicin in killing cancer cells, but it has less high side effects. That is important as well. And it is a magic that we just put some doxorubicin inside a particle, and then we basically strike out one of the major side effects of it. So when will it come? It is already there. How there are many challenges. If the question means that when will you be going to a pharmacy with a prescription and buy it like a paracetamol tablet, I think there are many, many challenges over there. We have to do many more webinars to actually go through them. But if you go for parenteral administration, it is already there. But it is not in that extent that we would like to see. That's my answer to the question.
Yeah. So any more questions from the audience? You can raise your hand or you can type your questions. Okay. So as we wait for the question, so just had a question like you being a doctor. So what do you feel about do you feel that the COVID vaccines, like some of them are kind of in nano form, they're the success stories in our generation? Like, what do you feel about it?
I think nano vaccines. There was a big hype, and when I entered this field, especially in the European circuit of research, there was a big hype, and there is still continues to be a hype about nano vaccines. I see that hype kind of becoming sluggish now. I don't know why. As far as COVID-19 is concerned, I think we still have to wait for concrete data. A lot of data has been put to larger audience, but I think they need more scrutiny before we can actually start drawing conclusions. My personal belief is it depends on the route of administration of the vaccine and what type of vaccine. I have taken vaccine myself, but they were not particulate vaccines. Particulate vaccine, it will depend on the route and the different type of biological barriers. Once again, that is where the physiological barrier actually becomes important. What kind of barrier you are trying to negotiate? And that is because the particles have to cross that barrier. Unless it doesn't cross. And this is very interesting because if you look, for example, to the SARS-CoV-2 virus, it's a nanoparticle. I mean, the average size of a COVID-19, what actually we found, the transmission electron microscopy, the size were somewhere around 80 to 120 nanometers. So we can consider them to be kind of a viral nanoparticle. Now, these viral nanoparticles, they can cross mucous barrier at ease. And that is why COVID is so infective. It can cross the mucous barrier in respiratory tract at ease. Because that is how it reaches the epithelial cell layer, and then it can bind with the ACE2 receptors. That's the pathogenesis, that's the remarkable ability to negotiate with the mucous barrier. Unfortunately, we don't have that kind of a leeway as a chemist. Our particles don't move like viruses, and we still don't understand why. So, unfortunately, Shomik, I don't have a yes or no black and white answer for you. Okay. I think we have to wait. A lot of data is there. A lot of data refuting it is there. Let's not get into too much controversy.
Sure. So there is a question from Professor Tarak Karmakar, a colleague of mine from the Department of Chemistry, IIT Delhi. So his question is like, can functionalized nanoparticles with proteins, carbohydrates, etc., mimicking a virus capsid help in a nanoparticle insertion into the cells? Like
very good
the barriers, and synthesizing such nanoparticles, functionalized nanoparticles is, however, a non-trivial task. So what are your comments?
Well, first of all, thank you, Professor Tarak Karmakar, for this amazing question. I think this is where chemistry and playing around with the surface chemistry becomes really important. Now, actually, we are already doing it. So if you remember that liposomal construct that I showed you, we are trying to functionalize surfaces with different hydrophilic chains. And it is coming from the viruses. So we have learned or tried to learn, at least in this space, from the nature. What we have seen is that, as I said, the viruses can cross the mucous layer at ease. In fact, it can cross most of the physiological barriers at ease. Now, why? Why it can cross? Now, there is no clear-cut answer. But what we have seen, especially for mucus permeating nanoparticles or viruses, that they have a very well-defined density of surface charge on the surface. And that is a hydrophilic polar surfaces that these viruses have, enables them to carry a kind of a meniscus of water with them. And that meniscus of water actually kind of shields the particle in a kind of a water bubble. And that helps them not to interact with the mucin fiber and permeate through mucus. In my lab, I'm trying to mimic all these things because these are fantastic hypotheses, but how to actually check them? We are trying it. It's not easy. I am fully convinced, and that is where I would like to agree with you, that yes, we have to start wondering, rather than what to put inside the particles, to actually focus on the surface. And we need to work on the surface, like with all the materials you have said. Proteins, for example, glycobiology, using the knowledge of glycobiology with all the carbohydrates. Interesting feature is, whatever molecules you are suggesting, proteins, carbohydrates, they are polar molecules. So yes, I completely agree with you. I think this is the time to switch gears from encapsulation to surface conjugation. That's where the future lies, in my opinion, if we have to succeed. And all the molecules that you have stated should work. But they all have to be hydrophilic molecules, in my opinion, and that completely aligns with your hypothesis, because proteins, carbohydrates, they will be all polar molecules anyway. We need to make the surfaces hydrophilic, and that is where I am fully in alignment with what you have said. I completely agree. Okay, next question.
Yeah, I think next question is actually Gagandeep asked a couple of questions, but I think they have been discussed already. I'll come back to a part of that question later, but we can take Anjali's question, like what type of nanocarrier proved to be the best till now for drug administration Is it dendrimer, lysosome based, or quantum dots, etc., and why?
Very good question. Again, thank you. Thank you, Anjali. If you look into the approved list of formulations, and when I say approved, I basically mean US FDA approved. Now, different countries have different approving agencies. For example, in European Union, you have the EMA, that is European Medicines Agency. Now, if you look into the list of approved formulations by US FDA, most of them are of lipidic constituents and are liposomes. So if you look into the current prototypes, liposomes are definitely leading the race, which has actually worked. Doxil itself is a liposomal formulation, which is the kind of a success story that in research world is circulated. Now, interestingly, if you look into what I said I have a lot of hope for nanocrystals. And you will be surprised to know that more than 10 formulations, which are nanocrystal formulations, have been approved by US FDA. And this has mostly happened in the last decade. So nanocrystals are really emerging fast, and they are emerging fast because of certain inherent facilities with them or strengths that they acquire. Nanocrystal basically means you are having the drug itself and making crystals out of it. Now, that has a lot of advantages. You're not using any excipient, you're not using any other additive. Your reaction is usually simple. I'm not saying always, but usually simple. So you're dealing with very little amount of reactants that already makes purification easy. The yield improves. You can even do kind of surface functionalization. And these crystals are known to dissolve very well inside human body because of 37 degrees centigrade temperature, different enzymes. So these crystals kind of snaps immediately in the bloodstream and you get 100% dose. So your encapsulation efficiency is 100% because it's basically pure drug with some surfactant here and there. So that is why more than 10 formulations have been approved very fast over the last decade of nanocrystals. And I'm not a clairvoyant person, but if you ask me, my forecast is, or at least my hope is, that in next 10, 15 years, the number of approved formulations of nanocrystals will supersede that of liposomes because there are certain inherent advantages with it. But as of now, that we are talking on 25th of August 2022, liposomes are leading the race. Dendrimers are still working. Now, there is one dendrimer formulation that has been approved in Australia. Not in other places, not to my knowledge. But there is a dendrimeric formulation called Viva Gel that has been approved in Australia. I have no idea how successful it is. It is not a breakthrough material, otherwise I would have heard. But it is at least approved in Australia. It is I think, for genital tract infections in women, it is approved for topical formulation. But other than that, I'm not aware of any dendrimeric formulation being approved so far. Quantum dots, difficult, because quantum dots are toxic and there is a general perception in research community that they are toxic. So the safety can be compromised. That is why quantum dots I don't see. And secondly, quantum dots are very difficult to load with drugs. You can do some surface conjugation, but to produce a therapeutic effect, you need to load these particles with drugs so that you can have enough material reaching there. Now, quantum dots, I'm not very sure. However, quantum dots are very good for imaging. And if we can somehow curtail this toxicity, which once again comes from the surface, so a way can be to passivate the surface by grafting it with different polymers, again, makes things difficult, but it is possible. I think quantum dots are fantastic materials for biomedical imaging, and we should work on it. I think quantum dots have been underrated because of its toxicity, but for imaging purposes, not for therapeutic purposes, for imaging and diagnostic purposes, quantum dots are great. But we can have a different webinar or we can talk sometime about this knowledge, quantum dots, but not from therapeutic or drug delivery perspective. From imaging perspective, quantum dots are great. And if we can passivate its surface, curtail down or bring down its toxicity, I'm very hopeful about them. Next question.
So we have a question from Astha.
Yes.
So she asks that once a nanomedicine is FDA approved, if any further modification in the nanomedicine is done by the owners, then is it still called by the same name? For example, Abraxane was approved in 2005, 2006. So there may be certain changes done in the formulation over 17 years. So are they still called by the same name? So that's the question.
I'm not actually sure, because this is this approval, and I'm sure there is some patent which usually leads this type of approval. These are very legal terms. What usually I have seen with pharmaceutical companies is once they patent a product, and when they keep changing it, even Doxil has changed to a certain extent. Every nanomedicinal formulation changes. They keep on producing supplementary patent applications so that they block everything, so that their competitors cannot go and actually catch them up over there. I personally think, I have, honestly speaking, I don't know actually, because these are legal materials and there are a lot of, I think there are places for interpretation. My own take is you have to go to that agency again and file for with more data, because the moment you are changing the, unless you, changing the configuration or the design of the nano formulation, it depends how much you are changing. If you are changing a lot, and don't ask me what is lot, I don't know, they have to decide them, then it has to continue with further patent application or preservation of intellectual rights. So you are allowed, I'm sure you are allowed, but there are certain procedures that has to be followed, and it will be depending on how much change you are doing. Now, what actually makes how much, that is something I cannot say. I personally believe if you change the fundamental design of the particle, then that will be changing. So it's a liposomal formulation, for example, in case of Abraxane, you have said, for example. So you're basically binding albumin to something. The problem is, say you change albumin with a different protein, I think that will be a different product. But if you just attach albumin with a different type of albumin with a different molecular weight, then the process will be simpler. Although I think you have to notify FDA that what the changes have you made. You have to be very clear and honest about it, and they will decide if it is a different product or not. So it's a kind of hazy. It's a legal document with a lot of discussion, and no clear-cut boundary is known to me. It depends the change that you are making. If you're making too much, then it's a different product. If you are just making subtle changes, then you can still go away with it with simple information. Okay? Next question, if any.
Any more questions? So actually, there is a question from Gagan, but I think mostly it is answered, but a part of it can be asked still. So what suggestions would you give to budding researchers working in this field? That's this question.
Many. Looking at my frustrations, you can understand I can give many suggestions, but let's not get there. Stay humble. The true enlightenment of knowledge is to know how little you know. Never get disappointed. Research is a journey. There is nothing called success or failure here. You keep trying. That's what I have learned. It is very important to discuss, and I think I don't know about I have hardly worked in Indian academia, so I have very little or in fact no experience. But in Western academia, I think I have also covered it here. Collaboration is very important. Academia is an intellectual job. We need to discuss with each other. We need to sit down. And I think too often I have noticed people just get into the lab and start doing stuff rather than actually having an intellectual analysis of it. It is very important to sit down with your colleagues and discuss, which is important. And another thing I would like to say, think about it. Just before you get into an experiment, design it, think it, criticize it. Don't depend on others to criticize you. criticize yourself. Self-criticism, self-analysis is the best thing. That's what I have found. Respect your seniors. Develop a habit of reading good literature. Make a habit that you will read at least one paper every day. That is very, very important. And learn to tell stories. Stories are important in our profession, that is academia. Someone told me publication is our currency. So publication and presentation, and to explain your research, explain your perspectives, staying humble, respecting seniors, reading and get acquainted with whatever is going on in your surrounding, and keep upgrading yourself. There is no space for complacency. You have to keep upgrading every day. When I started in Edinburgh 2006-7, and that time nanotoxicology was a big hype, and the way I started, and after in 2022, I see it's basically backdated. New information has arrived. I mean, I have to keep upgrading myself and everyone. I mean, Shomik is sitting here, all the other faculty members are sitting here, you are sitting here. We all have to change, upgrade ourselves, and there is no end to it. Stay hungry, keep exploring, listen to your heart. I think that's pretty much sums it up. Any other questions?
Okay. More questions? Don't see any hands raised as of yet, and also no more text. So okay. So I think
I see Gagandeep Singh has sent a thank you message. Thank you, Gagandeep. I hope I could enlighten you in some way and inspire you and motivate you.
Okay, so I think if you have if you have further questions, you can always write to Dr. Bhattacharjee, and I hope he will answer all your questions. And yeah, I think if there are no more questions, then let's thank Dr. Bhattacharjee for sparing some time and giving this fantastic, very insightful lecture, and also for the discussions. And I would like to thank all the participants also for their very enthusiastic questions and active participation. As always in this
Thank you, Shumi. Thank you, audience. You have been a nice audience. Some nice questions. But listen, I'm a very mobile savvy person. So I'm always connected 24/7, 365 days a year, 366 days on a leap year. So just you have my email ID, or if not, just get it from Shomik. He's most of the time a partner in crime of me. And any question, just fire it up, and I will try to respond as much as I can. Because I think these discussions are important, and rather than just going into lab, we should sit down and exchange ideas. That is how the old Greeks did it. We should do it also. Thank you. Thanks for everything.
Yeah, and before we just leave, I would like to thank a few people, like the committee which organizes these Pratidhwani talks, especially Professor Janaki, who helps me in organizing this. And also would like to acknowledge the contribution of our HOD, Professor Kurur, and other mentors who have supported us in organizing these talks. So thank you everyone once again, and thank you Professor Bhattacharjee again for joining us today.
Thank you. Have a nice day or nice evening in India. Bye.
You too. Bye-bye.
Bye. I'm logging off. Okay. Bye.